Now we're going to talk a little bit about this processing that occurs in mRNA in eukaryotic cells. Remember, the first thing that happens to the mRNA once it's produced through transcription is this process called splicing. What happens in a eukaryotic cell is that the protein code that comes from the gene is not one long continuous sequence. Instead, it's broken into fragments. It contains what are called exon portions and what are called intron portions. An exon is actual protein code. It's a sequence of bases that will be decoded by the ribosome during translation. An intron is different. An intron separates the exons from each other. It's also just a sequence of bases, but it doesn't code for the protein. So, what has to happen during splicing is the introns in the mRNA have to be removed, and the exons have to be brought together and joined. And again, that's this process that we know of as splicing. So on this slide, you can see a drawing of a gene in the DNA, what we refer to as the primary or the immature RNA that comes from that DNA, and then the final mature RNA that's produced after processing. So if you start at the top here, they're showing us a gene in one of the strands of the DNA, the template strand. You can see that part of this gene is regulatory and part of it codes for protein. So the regulatory area is listed as the promoter. That's where the RNA polymerase enzyme is going to bind. The sigma is going to bind. And then the core is going to transcribe all the way down the protein coding region of the gene. And what we will get produced from that is this immature mRNA. You can see that the whole protein coding region of the gene is now recorded, if you will, as an immature RNA molecule. So the exon regions are in it and the intron regions are in it. It's the exon regions that actually contain the code that's needed to build the protein. The intron regions don't contribute to that code, so we need to remove those. So during splicing, these lighter colored regions are literally going to be cut out of this RNA molecule, and then the darker colored regions, there are 3 of them, are going to be brought closer together and attached. So we have one continuous mature mRNA molecule. Now, this splicing process is fairly complex. It actually is achieved through not one, but a group of proteins and small RNAs that are called small nuclear ribonucleoproteins. It's a very long name, a very long term. We usually shorthand it when we talk about these as SNRNPs, or what is affectionately referred to as SNRPs. So this is a big complex of protein and RNA. And the RNAs are small. They're not big, long, complex RNAs. They're little short sequences of RNA. And what you end up with, this combination of protein and RNA, is referred to as SNRPs. The RNA portion of the SNRPs is the catalytic portion. Remember, we got to do some work here during splicing. We've got to cut through phosphodiester bonds. We've got to break them. And then we've got to build them again between the individual exons. So anytime that you have a catalytic RNA, you can give that RNA a name. You can call it a ribozyme. It's not an enzyme, because remember, by definition, an enzyme is a catalytic protein. A ribozyme is a catalytic RNA, and SNRPs are catalytic. So this whole massive combination of protein and ribozymes is referred to as the spliceosome. The spliceosome. That word translates as splicing body. In this drawing, we can see this splicing process begin. Here's our immature mRNA shown in yellow. And in this drawing, it has 2 exons, the darker areas, and 1 intron, the lighter area. And what they've drawn in behind the mRNA are these purple molecules. These represent SNRPs. These are a combination of protein and RNA. Now the SNRPs are going to come together and form this big complex called a spliceosome. And essentially what the spliceosome does is it brings the 2 junction points between the intron and the exons. It brings them together and it creates a loop out of the intron. Now, they've given us some details over here about the fact that there's an adenine base in this region and there's a hydroxyl group that gets recognized. Don't worry about those details. Just know that the spliceosome is capable of moving the immature RNA in such a way that it creates a loop out of the intron and it draws the 2 exons close together. It can then break the RNA strand so that the intron region is removed, and then it can bring the exons together and bind them. So in the end, the intron is excised, it's removed, it will be recycled, and the exons are brought together and joined. Now remember, in a eukaryotic cell, splicing is only part of the processing that goes on. The other things that have to happen is a cap has to be added to the RNA and a tail. These are the terms that we use. What the cap is a molecule called 7-methylguanylate, or what we shorthand as M7G. And that M7G has 3 phosphate groups attached to it. The cap structure is going to provide some protection for the RNA molecule against degradation. Remember, we have said before that RNA is not a particularly stable molecule, and mRNA in particular is not going to spend a lot of time in a cell. It will be translated to produce protein, but then it will be fairly rapidly degraded and recycled. So the cap that gets placed on the mRNA molecule is going to help protect it from degradation just long enough for it to be translated. The tail is a little bit different. The tail is there as part of the regulatory mechanisms of translation. And the tail is actually composed of a long repeat of adenine bases. Somewhere between 100 and 250 individual adenine nucleotides get added on to the end of the mRNA as a tail. Sometimes you'll hear it called the poly-A tail. And again, it's there as part of a regulatory mechanism. So on this slide, we can see a drawing of this now mature RNA molecule. you can see that the dark yellow region in the center, this is where the actual code for building a protein is. There is going to be a little bit of sequence. There's going to be some bases, some nucleotides on either side of that coding region that are not part of the coding, not part of the sequence of bases that help build the protein. So they're referred to as untranslated. They're part of the RNA, but they're not actually going to get translated to build a protein. And then outside of those untranslated regions, we see the poly A tail on the 3' end of the molecule, and the cap structure, the M7G with the 3 phosphate groups, protecting that mRNA, at least for a while, from degradation.